A-Level Biology 9700, Paper 4, Variant 2, May June 2025. And in this video, we will discuss the question number 5. And question number 5 is a number of diseases in humans can be treated using recombinant human proteins. These these are produced by recombinant DNA technology. To produce a human protein for treatment of a disease, recombinant DNA technology needs a gene coding for the particular human protein. Outline the different ways that can be used to obtain a gene that codes for a human protein. So here in this, we have to discuss the ways by which uh, a human protein we obtain a gene that codes for human protein. So first of all, they have talked about the recombinant human insulin. So recombinant DNA is DNA that has been formed by combining genetic material from two or more different sources. So recombinant DNA, we have to outline the different ways. The first way which we can discuss uh, is that the extraction from genomic DNA, that is, the gene can be extracted or cut out directly from the complete human genomic DNA, and then restriction enzymes, endonucleases, are used to cut the human chromosome at specific recognition sites, theoretically flanking the desired gene. And then we can say the second method can be we can use the reverse transcriptase to create complementary DNA. So this is the most common method, especially when the final host is bacterium. So it is yield genes without introns. So messenger RNA is first isolated from human cells that naturally express the protein, so the messenger RNA is already processed and has no introns. So the enzyme reverse transcriptase uses the messenger RNA as a template to synthesize a complementary single strand of DNA. Then the single-stranded complementary DNA is then converted into a double-stranded DNA molecule. So in this way, the resulting complementary DNA contains only the coding of the genes, making it directly ready for expression in bacterial other host cells, thus avoiding the need for RNA splicing. So the third method which can we use is the if the amino acid sequence, this is the gene synthesis, you can say that if the gene amino acid sequence of the protein is known, the corresponding DNA base sequence, codon sequence can be determined using the genetic code. So the gene is then synthesized chemically from scratch in a laboratory using automated DNA synthesis machines. So in this way, we can say that this method allows for optimization of gene sequence. So another method, we can say that the amplification using polymerase chain reactions, so a small sample of the desired gene can be quickly produced in large quantities. So in this way, these are the different methods which are different ways that can be used to obtain a gene that codes for human protein. So I would like to write all these one by one on the given space. So I will start with the first that we can extract from genomic DNA. So the gene can be, I would like to, the gene can be extracted or cut out directly from the human genomic DNA restriction enzymes are used to cut the human chromosomes at specific recognition sites. Then the next method is the messenger RNA. The enzyme reverse transcriptase reverse transcriptase uses the messenger RNA as a template to synthesize two complementary single strand of DNA. The single stranded DNA DNA, complementary DNA. C stands for complementary, is then converted into double stranded DNA molecule. So the third method which can be used is the if the amino acid sequence of protein is known, the corresponding DNA base sequence can be determined using the genetic code. So it was all about the different ways by which uh, can be used to obtain a gene that codes for a human protein. Now moving to the next part of this question, which is diabetes mellitus is a disease in which the blood glucose concentration cannot be controlled. So many people with diabetes mellitus use recombinant human insulin to help control their blood glucose concentration. Before the recombinant human insulin became available, animals were the main source of insulin. So explain the advantages of using recombinant human insulin to treat diabetes. So the advantage of using recombinant human insulin are because we can, in this way, we can reduce the allergic reactions in humans, and then secondly, we can have consistent and reliable supply, and third, we can have higher purity and safety. You can see safety from different type of diseases. So if we now I will write all these 3 points in detail. So we will start with the first point, that is the recombinant human insulin. Insulin is identical identical to the insulin naturally produced by the human pancreas animal insulin contains small differences in amino acid sequence compared to human insulin, which can trigger an immune response or allergic reaction. Secondly, the supply of animal insulin was limited by the availability of animals this method is highly scalable ensuring a consistent reliable life. So the production process avoids the use of animal tissues, eliminating the risk of transmitting animal disease. The production process avoids or eliminates the risk of transmitting animal diseases. So these were the points which we can use to write as an answer. Now we come to the next part C of this question. Explain why the DNA involved in the production of recombinant human insulin is termed recombinant DNA. So the recombinant DNA is DNA that has been formed by uh, combining genetic material from two or more different sources. So in the production of recombinant human insulin, the first source is the human insulin gene, which codes for the insulin protein. The second source is typically a bacterial plasmid. So the term we use because the human gene is cut and then inserted into the bacterial plasmid using enzymes like transcription endonuclease. So I would like to write the answer. Recombinant DNA is DNA that has been formed by combining genetic material from two or more different sources. As for example, the human insulin gene and the second bacterial plasmid. So it was about part C. Now we are moving towards the last part of this question, that is D. And let's see what is in D. So the recombinant human insulin and these are insulin proteins that have been slightly altered amino acid sequence compared with recombinant human insulin. So these analogies can be more effective than human insulin. Synthetic genes coding for insulin analogies has been developed. The bacterium Escherichia coli can be used as a host for synthetic gene for the large-scale manufacture of an analog. So when scientists have determined the changes that are needed to produce an insulin analog, they can uh, obtain a synthetic gene coding for the analog by making changes to a length of DNA using genetic engineering. So, suggest how scientists genetically engineer a synthetic gene coding for the insulin analog and explain how the changes that make allow the correct analog to be produced. So we have to suggest that how scientists genetically engineer a synthetic gene coding for the insulin analog and explain how the changes that make allow the correct analog to be produced. So we have not only to give suggestion, but we have to explain it as well. So uh, as an answer, we have to discuss this it in detail. So the first you can see that is the we can use the genetic engineering method. So scientists typically use a process called site-directed mutagenesis, or you can say uh, they use few bases in a non-DNA sequence, or they precisely change a few bases in non-DNA sequence. Then scientists first identify the desired amino acid substitution, as for example, changing alanine to glycine that will make the insulin analog more effective. Then they then determine the specific codon in the original insulin gene's DNA that code for the unwanted amino acids. Then they synthesize a short single strand piece of DNA that is complementary to the target region of the insulin gene but contains the one or two mismatched bases needed to change the codon to the one coding for the new desired amino acid. So thirdly, they amplification and insertion, these primaries used in a polymerase chain reaction, PCR, so like process to copy the entire plasmid DNA containing the insulin gene. So during this copying, the mutated primer is incorporated, creating a desired change. Then the second part of this question is the how we can explain how changes allow correct analog production. So the changes made in the DNA sequence directly affect the subsequent processes of gene expression. So during the transcription, the genetically engineered synthetic DNA sequence is transcribed into a messenger RNA. So change in the DNA base sequence results in a corresponding change in the sequence of codons on the messenger RNA. So the altered messenger RNA is then translated by the E. coli ribosome. So because the scientists changed the DNA codon, the messenger RNA codon is also changed. So the messenger RNA will leading to the transfer RNA bringing a different specific amino acid to that position during protein synthesis. So the final polypeptide chain produced will have the correct slightly altered amino acid sequence required to make it more effective than natural human insulin. So this is based on the principle that the genetic code is triplet code, where a sequence of bases dictates the sequence of amino acids. So this is it means this question consists of two parts. The first is that we have to uh, suggest how scientists genetically engineer a synthetic gene for an insulin analog. So I'm going to write firstly for this question answer. So scientists typically use a process called site directed mutagenesis. To precise, precisely change a few bases in a known DNA sequence. Scientists first identify desired amino acid substitution. That will make the insulin analog more effective. Then determine the specific codon for the unwanted amino acids. They synthesize a short single strand piece of tape. Piece of DNA that is complementary to the complementary to the target region of the insulin gene. So primer is used in polymerase chain reaction to this. Then the second part of this question is the explanation of how changes allow correct analog production. So for this purpose, the changes made in changes made in the DNA made in the DNA base sequence results in a corresponding change in the sequence of codons on the messenger RNA, which is then translated by the ribosomes. The polypeptide chain produced will have the correct slightly altered amino acid sequence. Required to make it more effective than natural human insulin. So it was all about this question 5. Please like, share, and subscribe my channel to watch more videos on these papers for your exams. And thanks for watching.